Good afternoon and welcome back to the 6th annual patient-driven precision medicine conference at Harvard Medical School. We are now in the process of the penultimate panel, which the topic is, well, if there's such a thing as ultra rare diseases with hyper-individualized therapies, how many common diseases and to what extent are common diseases themselves very individualized, very separate, but similar manifesting diseases? And to what extent are therapies that are very targeted going to be effective for diseases that we don't think of as rare diseases? Before I go on to the panel, I would be remiss if I did not note that the very interesting slides that you've seen in their interludes that show various graphics of studies of COVID-19 are courtesy of the Galenborg Lab and Sehi Lee, who have done very nice work in that regard. So we have another star panel for this discussion. We have first, let me just make sure I have the right order. Yes. First, we have Pradeep Natarajan. Dr. Natarajan studies the genetic drivers of human atherosclerosis, a very common disease, using genetic epidemiology and large-scale sequencing studies. He also studies digital health and genotype-driven human investigation. Roman Yelensky is the Executive Vice President of Bioinformatics at Gritstone, a impressive company that focuses, and well, his focus is on the discovery of neoantigens for use as cancer vaccines. Prior to that, so that's a very much, on the one hand, very hyper-individualized, yet it's for a common disease, cancer. Previous to that, he was vice president at Foundation Medicine, where he co-led the development of the Foundation One product. For me, the most impressive feature is he received a PhD from our Bioinformatics and Integrative Genomics program at the Division of Health Sciences and Technology of Harvard and MIT. Less impressively, got a master's from Stanford. We then have Dr. Stanley Shaw, who is the inaugural Associate Dean for Executive Education at Harvard Medical School. And he's developed a really interesting set of programs for companies and executive leaders who are interested in doing research in healthcare, getting more involved in healthcare. And I've seen a few of these and heard feedback. People love it. He's also involved with, he's Chief Scientific Officer of One Brave Idea at Brigham and Women's Hospital, and he co-founded the Center for Assessment Technology and Continuous Health at Massachusetts General Hospital. He also was part of the team that developed the first research kit iPhone app in partnership with Apple. He's also happens to be a great scientist and has a more impressive than him spouse, Dr. Alice Shaw, who we unfortunately could not get to come, so we just accepted him instead. And his children are really good at many things, including the piano. And it is because of Stan Shaw's better half that we've come to know Linnea. Linnea is a mother, an artist, a writer, an adventurer. She refers to herself as a terminal optimist, which is a bizarre but funny sense of humor in describing the fact that she's been diagnosed for over a decade with lung cancer, never fully cleared the cancer from her chest, and has gone from one phase one therapy trial to another, and is unbelievable, proof of which you'll see her mask on her face because she wants to protect herself and other patients at the hospital. So with that, why don't I let everybody start with their first presentation. Pradeep, over to you.
Great, thanks so much, Zach. I'm super delighted to be engaged by this group and with you, and thanks to all for organizing. You know, as Zach highlighted, the primary focus of my research is to understand the root drivers of atherosclerotic cardiovascular disease. You know, this is the disease process that encompasses coronary artery disease, the root cause of myocardial infarction, as well as stroke. So the leading cause of death in the US, and the dubious distinction of now being the leading cause of premature death worldwide. So on the clinical side, I direct what's called preventive cardiology, where we try to understand the causes for a given individual for, or at least risk factors for a first heart attack. And the folks who come to us are people who often don't fit within neat bins. So we use genetics, you know, within the clinic and then separately on the research side, to try to understand individual risk factors and then try to understand whether those risk factors can be used to actually predict specific therapies. And you know, much of the research that we're doing is fairly early observational work, but I think forms robust hypotheses to be tested prospectively that we can discuss. And I'll share with you a couple of these stories. Two examples, one where we're looking at germline genetics and one actually on somatic genetics. So, you know, you may have read a lot in the lay press and on social media about polygenic risk scores to leveraging genetic association studies to predict risk for common diseases, including coronary artery disease, which, you know, most of them have some degree of heritable basis. Those genetic association studies, the data in totem, can be captured into a single score. And that, in addition to clinical risk factors, can be used to refine cardiovascular disease risk. A surprising observation that we made a few years ago was when looking at randomized controlled trials of statin versus placebo. As you know, statins are tried and true medicines to prevent the first onset of a heart attack as well as recurrent risk. Surprisingly, we observed that individuals who were specifically genetically predisposed to coronary artery disease to a high degree had a greater absolute and relative benefit from the statins. And why this was surprising is that pretty much in every clinically defined subgroup analysis of statin randomized controlled trials, the relative benefit was the same. Meaning, for the amount that you lower LDL cholesterol, the relative amount that you benefit from cardiovascular disease risk reduction is the same. And the reason why we give people who are at higher risk statin medicines, you know, diabetics, hypertensive, smokers, is purely because the absolute risk is higher. And so you maximize the absolute risk reduction just because they have more risk factors. What was surprising here is that actually you maximize the absolute risk reduction not only because they were at higher risk, but they had a greater benefit for the amount of lowering for LDL cholesterol. And there's a fair amount of controversy for middle-aged adults, you know, people who are asymptomatic, lots of folks who come to the office on whether they should be on a statin medicine. And we defined a framework that actually, using observational data within healthcare-associated biobanks, suggested that about 1 in 25 asymptomatic middle-aged adults where there is clinical equipoise whether they should be on statins, actually knowing their genetic predisposition to coronary artery disease can help influence whether this should be prescribed. So just to recap, within randomized controlled trials and post-hoc analyses, we see a greater benefit, relative and absolute, for those at high genetic predisposition. And within 3 healthcare-associated biobanks, so Mass General Brigham, the University of Pennsylvania, and Mount Sinai's biobank, there was a large number of individuals who had high genetic risk, obviously not recognized by current clinical paradigms, who we think may benefit from having this information. Separate question is whether you should understand this information earlier, and we can talk about that in the panel. Now, very quickly, I'll just pivot over to the second part. In our large-scale human genetic analyses, we derive the DNA from circulating white blood cells. Now, using separate statistical methods, we can actually detect the presence of acquired mutations from DNA in those white blood cells. It turns out as people age, they have an increased prevalence of mutations in leukemia genes without actually having overt leukemia, meaning adults greater than the age of 70, about 1 in 10 have this phenomenon. It's, you know, termed clonal hematopoiesis of indeterminate potential. A few years ago, we made a surprising observation that these individuals were actually at greater risk for cardiovascular disease. Mouse models, as you see here on the side, show that there's also a greater burden of atherosclerosis in mice who are modeled to have clonal hematopoiesis. Now you could say, treat them similarly, they have higher risk, treat them with a statin. Turns out, at least first in mouse modeling, if you inhibit a very specific pathway that has been known to be implicated in cardiovascular disease, the IL-1 beta pathway, you actually specifically inhibit atherosclerosis to a greater degree in mice that are modeled with CHIP. This is with the inhibitor here. And we used kind of an extension of Mendelian randomization to model that therapeutic effect. For individuals who develop clonal hematopoiesis, if they were naturally deficient in signaling through this pathway through a coding SNP, were they at greater benefit for lowering cardiovascular disease risk? And it turns out individuals with clonal hematopoiesis, this is, again, one in 10 individuals who's above the age of 70, had a much more substantial risk reduction. And this is a medicine that's not being conventionally considered in clinical cardiovascular disease practice. And so we think this could actually be kind of really the first paradigm of a novel therapeutic target for a molecularly defined subgroup for cardiovascular disease or for coronary artery disease.
Thank you. So now let's move on to Roman. Why don't you give us your intro?
Thanks very much, Zach. And it's a real pleasure to be here at the conference. I've been enjoying the conversation and the discussions all day. Really important topics. So as Zach introduced, I'm the Chief Technology Officer of Gritstone Oncology. So Gritstone Oncology is a biotechnology company. We're about 5 years old. We are headquartered in the Bay Area near Berkeley, but we also have a significant site here in Cambridge near MIT, which is the site I lead our informatics and genomics work. And Gritstone is focused on cancer immunotherapy and particularly on cancer immunotherapy using neoantigens. And I'll give a brief introduction of what that is and how this is actually a hyper-individualized therapeutic approach for cancer, obviously a common disease. So I'll try to share. Okay. So hopefully you're able to see my slides. And so the cancer immunotherapy of solid tumors has really experienced a revolution over the last 5 to 8 years. And what we started to see in solid cancers was cancer immunotherapy are really dramatic clinical benefits for some patients with so-called immune checkpoint inhibition. So this is just an example. There's a PET scans here, a before and after of a patient with non-small cell lung cancer treated in one of the early stage clinical trials of one of these immunotherapies. And you can see where on the PET scan here in the top left corner of the slide, really widely metastatic disease, a death sentence for this patient. And with the patient then receiving, in this case, it was atezolizumab, one of the PD-L1 checkpoint inhibitors, experiencing not only a rapid elimination of the lesions, at least by PET, but also actually a reduction in lesions and that resulted in long-term survival. And in fact, this patient is still in clinical remission. This kind of outcome for a patient with advanced metastatic solid tumors is really was really unthinkable before this kind of therapy. And the question is, how does this kind of how can this kind of outcome, which is actually quite rare still, even with the therapies we have today, can be extended to more patients? And what was learned when the genomics of patients with advanced solid tumors that were treated with immunotherapy and responded was studied, was that what the immune system was recognizing was the so-called neoantigens. And this is actually a relatively simple concept where if you have a normal protein that is not mutated in your genome, and that protein is expressed on the surface of tumor cells or normal cells, the immune system cannot recognize, sees that as self, and leaves those cells alone. Whereas on the other hand, if you have a mutation in the cancer genome, which can result in the presentation of a mutated peptide on the surface of the tumor cells on the HLA, that is so-called neoantigen, looks foreign to the immune system, can be recognized by T cells, and the tumor can be eliminated. And what was shown was that when patients, these rare patients that were really responding to immunotherapy, likely the principal target of that recognition was neoantigens. And then that raised the question of, well, then why do not more patients respond to cancer immunotherapy? And the answer is that, at least in part and in large part, is that most patients actually with advanced cancers either don't have T cells to neoantigens or don't have enough of them. So this set up about 5 or 6 years ago a therapeutic hypothesis of, for most of the patients with solid tumors that do not have enough T cells against their neoantigens, can we immunize with neoantigens to really provoke these T cell responses that can then go and eliminate the tumors? And that is the therapeutic hypothesis that Gritstone was founded on. And so the product we have in development in phase 1 clinical trials, it's called GRANITE. It is a personalized neoantigen cancer vaccine. So it is really a completely individualized treatment for a common cancer. The reason being that, in fact, neoantigens are usually private to each patient. So these foreign markers on each tumor really arise from random passenger mutations anywhere in the cancer genome. And so this neoantigen signature that potentially the immune system can recognize can only really be addressed for most patients with a personalized therapy. So our process for this personalized therapy is diagrammed on this slide, where the starting point is a routine clinical biopsy, where we then, that gets shipped to our labs in Cambridge here. In fact, we do a tumor exome sequencing, tumor transcriptome sequencing, as well as normal exome sequencing to identify the cancer mutations. Then from the large set of mutations that we may identify for that patient, it could be hundreds of mutations depending on the tumor type, we select the subset that is most likely to be immunogenic and likely to provoke that kind of immune response against the tumor that we want. And in our case, we delivered the 20 neoantigen candidates. And this we actually do with a machine learning artificial intelligence-based model that is trained on a large data set that we generated of HLA-presented peptides. Won't get into details on that. Then given the set of neoantigens that we've identified, we create a personalized vaccine for that patient, and actually 2 different vaccines, because what's been shown in the field with infectious diseases that actually when you deliver the same antigen with multiple modalities, you can provoke a stronger immune response. So we make a personalized adenovirus for that patient, as well as a personalized RNA, and we give these in sequence, the virus as a prime to really instigate the immune response, and then the RNA as a boost to keep that response at a high level to eliminate the tumor. And then what the physician gets back is an injection that he administers to the patient to administer in conjunction with currently used immune checkpoint inhibitors, PD-1, to then allow those T cells, once they're generated by the vaccine, to go to the tumor and attack and eliminate those tumor cells. So we are now in phase 1 clinical trials in dose escalation. We've made a number of these vaccines. We reported some early data earlier this year. Of course, we're generating a lot more data. And we see this as a potentially very exciting prospect for a impactful individualized therapy for common diseases, when we're looking now at lung cancer, gastric cancer, as well as colorectal cancer primarily. With that, I will pass on.
Thank you. So, Stan, sounds like complicated business. Are doctors ever going to understand that? Or are we all going to have to subscribe to your executive education programs? What's going on here?
Yeah, so well, thanks for the introduction and the invitation. I've been a fan. I think I've attended every one of your conferences, and it's a real honor to participate in a conference that really puts front and center the stories of patients and their families. So our writ for this panel, I guess, was to talk about education. As Zach said, I have kind of a broader view of education in terms of educating broader audiences across the healthcare industry. And I think, since we're talking about conceptually transformative changes in how medicine might be practiced, including in, common diseases, like cardiovascular disease, I think my thesis is that we're going to need education of different kinds for stakeholders across the ecosystem. Bapu, Jenna raised the issue before about value, and I think patients, physicians, health systems, payers, leading advocacy groups, and biotech pharma, they all have different concepts of this value. And so there's education about the technical aspects, but I think there's education about how this should play out in the real world and in real broad clinical practice. And I'll motivate this with an old historical example. So if you look at the adoption of X-rays, for instance, at the turn of the last century, so they were invented at 1895, and then you can see that the uptake took over 20 years. And this is utilization data from the University of Pennsylvania Hospital. And Diana Long, in her studies, highlighted a number of factors, which I think are still relevant to the discussion even of hyper-individualized therapies. First, there became a specialized role of a rechinologist, so that speaks to the training piece. There had to be economic change in that the person taking the X-rays would retain 25% of the fees generated by the study. And I think there had to be a broader cultural or system change in how people practiced and where the work happened. And so there was a time when people wanted healthcare in their homes, and hospitals really were a place for the chronically ill. And with the advent of X-rays to diagnose fractures, for instance, it really started to shift in hospitals being a place of acute diagnosis and acute treatment. If we look specifically at physician education, there's relatively little data that I could find, so I found a couple of surveys. One was co-sponsored by the National Organization for Rare Disorders very recently, huge outreach, and 1,500 practitioners responded, and more than half actually had 1 to 5 patients with a new rare disease diagnosis in the last year. So these were reasonably experienced clinicians. I thought it was interesting, 62% of this highly selected group still preferred to refer patients for a gene therapy discussion. And at the time, 63% were unaware of the 4 FDA-approved gene or cell therapies.
1 minute.
If you look at the broader field of DNA or RNA-based therapies, this is a survey of 280 or so clinicians. And as you go from dark green to light green, it scales from very familiar, familiar, somewhat familiar, limited, and none. And so you can see that oncologists do the best, pediatricians are quite good, but there's still very little awareness. And then let's stipulate that we can get those awareness numbers up. I just leave you with a slide from my field of cardiology and the current discussions. If you talk about statins, which are probably one of the most powerful, efficacious, common disease drugs that will ever be invented, this group looked at different kinds of vulnerabilities, whether it's age or racial makeup, female gender, lack of insurance, etc. And that line, as you increase the number of these vulnerabilities, you can see the decreasing percentage of statin use, right? And so if you just look at gender and race, Black men and Black women are approximately 80% as likely to be prescribed a statin when it's indicated as a white man. And so I think it's a reminder that even if we surmount, education of physicians, but also other stakeholders, there's a long road to hoe to make it effective in the real world.
Thank you, Stan. I don't know where to start, Linnea, because you have so many things you could say about what we just discussed. On the one hand, you have a common disease. On the other hand, you've had lots and lots of different, pretty specific therapies. How do you even think about that? I'll let you, since I don't have a good way of framing it, I'd love to hear your framing of it.
Now, I was thinking about that just as I was listening to everyone, how I do have a relatively common disease, but I have a rare form of a common disease. But because targeted therapies have been so very successful for my particular mutation, which is an EML4 ALK translocation, it actually has bumped the overall survival of all of lung cancer up, which is pretty stunning. And I should also add that I was actually diagnosed back in 2005. So I have been living with this disease for over 15 years now. And in 2005, there were so few options as far as treatment. So I was fortunate to be diagnosed at an early stage, had most of my left lung removed, followed by 4 rounds of adjuvant chemo, but my cancer came back right away. And radiation, unfortunately, was not an option because of the way my cancer presented. So we just watched and waited. And in the summer of 2008, I was restaged to 4, told I had 3 to 5 months left to live, actually got ready to die, prepared my family, said goodbye. And then they discovered that I had this oncogene, this driver in lung cancer. And that's when I entered my first phase 1 clinical trial for an ALK inhibitor. And currently, I am in my 4th phase 1 trial. This one is mutation agnostic. It's instead an anti-TROP2 antibody. And today, I just had my 4th round of that. And so that's why I'm in this environment. I just finished infusion an hour ago, but I'm so extraordinarily grateful to be here and to be part of this, be here in the big sense, but also today to be part of this conversation. Thank you.
But Linnea, I happen to know
Yes.
that you've made yourself even more exceptional. It was not enough that you had a lung cancer and not enough that you had this mutation that gave you perhaps this opportunity for a treatment. But as a result of treatments, you've developed many more mutations. So you're becoming a very, very rare bird.
Yes.
So how do you and your doctors think about, going from that common pathway to, okay, let's say a more rare version of cancer, and now you're super rare because you've accumulated a combination of mutations that, for all I know, you're unique. So,
yeah, honestly, it's tricky because, the benefit that I mentioned, that the overall survival has gone up. The median survival of my particular type of lung cancer is now, I think, I'm going to get this wrong maybe, but I think it's 6.8 years. And it has kind of bitten people like me in the ass because I have now been through 3 ALK inhibitors. There are no other ALK inhibitors currently that my secondary acquired mutations are sensitive to. And that's why I'm trying something that is mutation agnostic now. And at a certain point in time, I realized that there was not a lot of financial incentive to develop a 4th generation ALK inhibitor because the fact that I am a rare group, and as time goes on and people die, that rare group becomes smaller and smaller. So I feel, again, so fortunate to be here, but I am also incredibly frustrated that a clinical trial isn't more of a social contract, and that those of us who are outliers, that I wish that there was some sort of blanket trial almost to figure out why we're still here, because I think it's poor science. I think it's a poor investment. to take us so far and then say, I'm sorry, but we've done everything we can. And that's kind of the terrain I'm in right now. I'm very lucky that I have Stan's wife, Alice, on my side. In fact, as the meeting began, she was texting me, and I know she's going to do everything she can to find something else.
Well, that's great to hear that Alice responds to you. She never responds to me, and for all I know, not often to Stan either. No, she is wonderful, and well, you're lucky to have each other. I mean, what a great team. I can't help it, but this is unfortunately self-advertising. We do have a trial that you have participated in and that we're about to do the analysis of for exceptional responders who have graciously given us their blood and their DNA so we can try to figure out what the heck is going on and why you're going through a lot, but still you're extraordinary success. And so why is that? So going back to the rest of our panel, Pradeep, I could be wrong. I train in pediatric endocrinology, not cardiology, but my impression is there's still a big parallel. Most patients are treated by very, very common drugs, and the targeted ones are still a very small subset. What's your estimate? Do you believe that will shift, that there'll be more individualized therapy? And if there will be, what do you think is going to be the biggest driver of that?
It's a great question. Yeah, I mean, I think for those of us trying to find precise ways to treat cardiovascular disease broadly, I think we're all kind of want to be oncologists, where there is, clearly a genetic basis for oncologic disease that can be functionally characterized and therapeutically targeted. I think for diverse cardiovascular diseases, including coronary artery disease, there are multiple different inputs that include, well-recognized risk factors like circulating cholesterol, blood pressure, and type 2 diabetes and smoking. All of those which will continue to be addressed as they have been for decades. And the question is the residual risk. Despite the current therapies of statins for non-diabetics and aspirin in a very small select group of individuals, people still get cardiovascular disease. And the key is still, we're not that great at predicting who is going to get cardiovascular disease. And the therapies that we offer to prevent cardiovascular disease, as you highlight, are pretty much the same. And it's largely similar based on risk assessments for diverse cardiovascular disease. The main issue in my mind has been phenotyping, because I think we are, there's still a fair amount of discovery science to be done. The germline genetic phenotyping allows us to pinpoint causal pathways, but it's still fairly limited in its ability to identify people who are going to respond to specific therapies beyond, I'd say, examples of pharmacogenomics as it relates to specific antiplatelets. I think as we broaden how we phenotype people, I think that is where we will start to better understand the opportunities for precision medicine. And I like to think of myself more as a preventive cardiologist, more so than a geneticist. I think with the understanding that there are diverse inputs. You highlighted our new interests in trying to quantify lifestyle behaviors in the environment using digital health. I'm on clinical service right now on the cardiology consult service at MGH, and we have somebody admitted for heart failure because he couldn't afford to take his medicines and didn't know how to set up going to see a cardiologist. The therapies for that patient will be very different than somebody else who is taking the medicines and obviously still sick. So I think separately there are systems issues, but I think really going beyond the genetics, metabolomics, proteomics, transcriptomics, those things are being done, but those are largely done in small scale and in largely healthy populations. So I think my biggest plug, I think, really is the need for more discovery science first to be able to move into the precision medicine part. I think the example that I gave on clonal hematopoiesis, where it allows us to be oncology wannabes, I think really is a new discrete example where we define a subset of individuals that probably have all the similar pathways, but there is a particular pathway where you can address that associated risk that's not being addressed now.
So Roman, the company that you're the CTO of, Gritstone, has a very ambitious and exciting goal. And of those patients out there with these common diseases like lung cancer, is this going to be, even though you're looking at individual neoantigens, is this going to be is the market going to be less than 1%? Or do you think there are going to be far more patients who might benefit from this kind of approach? And if that's true, what are the main things getting in the way?
Yeah, great question, Zach. So the interesting thing that we learned about neoantigens and the likelihood of forming neoantigens, it's actually that neoantigens are a small fraction of the number of the mutations that actually occur in genomes, and it may be as little as 1 to 2% of mutations actually become neoantigens. So the size of the patient population, it really varies by indication to the extent that the indications vary by the number of mutations that the tumors within that indication may accumulate. So for example, in the situation of non-small cell lung cancer that's smoking related, not linked to one of the oncogenes, actually there could be hundreds of mutations in each of those cancer genomes. And so there, actually, we anticipate the majority of patients will have enough neoantigens, maybe 70, 80% of patients or more. There are a lot of cancers, as you know, that have this kind of intermediate mutation burden. Maybe there's only 50 mutations in the tumor or 75. And in that case, there's actually going to be a minority, but a significant minority of patients that have enough neoantigens for this kind of therapy. Maybe 40 to 50% of, say, colorectal cancer patients. And then there are some indications, at least from what we understand today, in the neoantigen classes that are readily accessible, arising from irregular point mutations, insertions, deletions, where those tumors have too few mutations for us to even consider. And some of the blood malignancies, for example, fall into that category. So it really does vary by the indication. The large solid tumors like lung and colon cancer, it's somewhere between significant minorities to majority of patients that may be treatable with such a therapy. Now, the challenge, of course, that brings us, exactly you point out, is that if the paradigm is proven to work, then you want to be making these kinds of vaccines for thousands, if not tens of thousands of patients. And we're obviously not there yet. We're in our phase 1 trials. And so we are treating, tens, and in the phase 2 trials, hundreds of patients, that range. So the current kind of manufacturing paradigms are, they're stretched, but they can accommodate such scales. To get to thousands of patients, we're obviously thinking about that already, planning for success, and that will require really dramatically more automation and scale. And the term is scale out as opposed to scale up. If you have an antibody drug in early stage trials, you'd make it in a small vat for your phase 1 or 2, and then you make it in a very large vat for a phase 3 and commercial. Here, a larger vat doesn't help you. It's a personalized therapy. So you basically have to duplicate these small vats over and over in a facility, obviously without linearly scaling everything else, because that becomes impractical. So that's going to be the challenge, how to make this in the thousands, which we're going to have to tackle in the future.
Stan, how do you talk to doctors or companies to navigate between a current state of the art patient who unfortunately probably will get standard therapy, and someone like Linnea who benefits clearly from not a standard therapy? What do you tell those doctors or those companies? What should we say?
Well, I think, by the time that a company has a product in, let's say, late or mid to late clinical trials, I think even rare disease companies are pretty good at working with, if there is an advocacy organization, and I think they have you know, an opportunity to hear about patient perspectives and what's meaningful, right? I think if you go to any of a number of patient-centric conferences, you will hear patients say, you know, we want to make sure that the outcomes in the trials are ones that are meaningful for us. And so if it's a hemoglobinopathy, maybe a rise in the hemoglobin count is less meaningful than the number of times I have to go in for a transfusion, or the number of missed days of work, or the number of missed school days for a child. I have faith. I think companies are actually quite sophisticated at getting those perspectives. Obviously, some do come to the medical school, and we love the opportunity to convene different perspectives, frontline clinicians and patients and their caregivers and clinical trialists. So those perspectives, I think, are meaningful. And, you know, on the physician side, I've had the good fortune to interview Linnea several times in our programs, and I can tell you she knows exactly her treatment-acquired mutation. She knows what amino acids were changed to what at what residue. Some of that may be from social media and the internet, but a lot of that is really just time and conversations with her physician. And, you know, to the extent that, you know, where you're at, you helm the Department of Biomedical Informatics, to the extent that digital health can maybe wick away time spent on medication reconciliation, maybe more time could, in theory, be devoted to these conversations and these educational conversations, because I think that gift of time, when you're talking about these difficult conversations, or even, you know, to talk about a clinical trial enrollment or consent, these are things that take a lot of time. And that's really a gift that hopefully technology maybe can help clear out and enable some more time. And, you know, maybe decision support can accelerate adoption, right? Once there's a standard, maybe decision support aligned with the EHR could help, you know, get our numbers up from, you know, suboptimal numbers of even prosaic things like aspirin and beta blocker after a heart attack. I mean, we're still somewhat fighting those battles.
Great, thank you. So this is now the audience part of the session. As before, while we wait for your question to accumulate, that will be questions will be presented by Dr. Kenneth Mandel from Children's Hospital and Department of Biomedical Informatics. While we wait that, we're going to exercise your Slido muscles by our trivia poll and then quiz. Rachel, please proceed.
And I will stop and move on to the quiz next.
Sure. No vitamin D true believers. All right, congratulations. All right. So, Dr. Mandel, I hope you're seeing some questions that you can share with us?
So let's jump in with the first question. And the question is to Roman. Great concept and technique. Could you tell us a little about the safety profile that has been observed so far from the vaccines you've developed, other than the efficacy profile you spoke about?
Yep, that's a very good question. So we reported earlier this year some very early safety data from 7 or 8 patients. And so far, the vaccine appears to be tolerated quite well. It is given in combination with immune checkpoint inhibition. So obviously those drugs also have their own safety profile, which is layered onto the regimen. So that obviously has to be considered. But the safety profile, at least from the data that we reported so far, is in line with what's expected for immunization and appears acceptable for this line of therapy.
Great.
Okay, great to hear that.
Okay, and the other audience question is to the panel. Could you please talk about the role of hyper-personalization across the lifespan of cancer? And maybe we'll see, even though Pradeep is a cardiologist, maybe we can generalize about hyper-personalization in chronic conditions that are common.
Yeah, and
I'll take
Pradeep, let me widen enlarge the softball that is being sent to you. So some people have known rare variants that put them at greater risk from childhood. So how do you see this lifespan going?
Yeah, I mean, so we have a little bit of a paradigm for this. There's, one well-recognized monogenic form of coronary artery disease, familial hypercholesterolemia, that afflicts about 1 in 300 individuals, pretty similar prevalence across ethnicities, readily recognized by significantly elevated LDL cholesterol. So often in the absence of genetic profiling, just purely based on severe hypercholesterolemia, we often will introduce statins at an early age, understanding that there aren't other risk factors than just high LDL. Now, with germline genetics for polygenic risk scoring, we found that actually there are more individuals that carry a similar risk. And based on the retrospective analyses, it seems like they also have a similarly greater benefit from statins. So we think potentially that those individuals could benefit for much earlier statin therapy. And it may not necessarily just be statins. And there's a lot of it, and the reason why folks come to our group is because they're younger and they want to know what they can do now to maximize their benefit throughout the course of lifetime. And our risk calculators are really predicting risk over the next 10 years, so they're not suitable for younger individuals. So we think that genetics may provide that trajectory of which it can be modified. And there may be a one-size-fits-all, or it may be specific therapies to be determined.
So just to, if you could expand on that a little more. So what change do you think there should be for patients who are assumed to have a common disease, but who have not had their, the fact that they have actually a common version of that common manifestation genetically? How much do you think practice will change once we look at common diseases and segment people by their omics into what may be a collection of rare diseases? How do you see that changing your practice?
Yeah, I think often what moves the needles are randomized controlled trials. Now, not everything in cardiovascular guidelines have undergone randomized controlled trials, and we don't practice within the framework of only using the evidence from randomized controlled trials. But generally, what will make strong arguments within guidelines are interventions that have undergone the rigor of randomized controlled trials. What is challenging for managing over the life course is that clinical trial can potentially be very expensive and be very long. Obviously, we don't want to wait 30 years before making some practice changes. So there is a fair amount that we do with the observational data, including in genetics, and leveraging ongoing clinical trials to identify high risk, high benefit groups. As Ken talked about, there's sense of value that's different across stakeholders. And in cardiology, as we expand to newer medicines, there's a fair amount of a three-way of the physicians, the patients, and the payer. And the value that comes down to is, are you able to get a sense of that patient benefiting to the most that you can for that specific medicine. And I think the way that we will be able to move that needle is as we more broadly profile folks, and that can be genetically, that can be other, and that is happening, within the biobanks and healthcare systems, here at partners, elsewhere. There's lots of direct-to-consumer genetic testing. So doing these clinical trials, 5 years ago was hard to do because these are tests that are not widely available. But now, patients within our healthcare system have this information that you can actually call them back in and do clinical trials informed by the observation.
Yeah, thank you, Pradeep. I think we are at time to give people the break. Zach looks like he wants to also say something.
Yeah, I just want to say one thing. Linnea, from where you're sitting, briefly, if you are a newly, God forbid, diagnosed a lung cancer patient, would you recommend extensive genome profiling or not? You're muted.
Absolutely. I mean, I think everyone should be. But, if we had more time to talk about this, again, as listening, I see the whole concept of personalized medicine and targeted therapies as being a bit of a misnomer, because as someone who has participated for over a decade in clinical trials, it's still a bit of a blunt hammer, this instrument. And I'm certainly hoping that as we have access to more information, that it truly can become personalized, even in the trial stage.
Great. So that's actually a vote for even more hyper-individualization. Thank you, Linnea. I hope you get to enjoy the outside today. Thank the rest of the panel. And thank you, Dr. Mandel. We regroup in 6 minutes. Thank you.
Thank you.